Hello there, and welcome to this Carbon Academy tutorial on AS level biology. In this video, we will look at cell immunity. This is part one of this topic, as there's a lot of content to cover, and it is quite complex. So let's look at the aims of this tutorial. So here are the aims. We'll explain the function of the immune system, define antigen and pathogen, list the main immune cells of the body and their characteristics, describe the two types of immunity, and explain how the adaptive and the innate immune system communicate, explain the importance of producing memory cells, and what the differences are between primary and secondary immune responses. So let's get started. The immune system is the body's defense system made up of immune cells, and certain immune cells patrol the body 24/7 looking for foreign antigens, just like the army. But what is an antigen? Antigens are proteins found on the surface of a cell. And all cells have antigens. However, not all antigens produce an immune response, such as the antigens of our own cells. However, foreign antigens do. Some common examples of cells that contain foreign antigens are pathogens, which are microorganisms that cause disease, tumor cells, i.e. from cancer, and transplanted organs from donors. Now here's a simple diagram of some of the cells that make up the immune system. There are many more, but we'll only focus on the four ones that are circled in red. Let's start with the neutrophils. Although you do not need to know this for A-level, it's important to know because it's the most abundant immune cell in the body. And approximately 100 billion are made daily, truly a staggering amount. The neutrophil functions as a phagocyte. And as usual in my channel, I like to explain the origin of the words. The word phago comes from the Greek meaning eat, and the word cyte refers to cells. So neutrophils are essentially cell-eating cells. Weird, right? But that's their function. They clear up dead cells and pathogens in our body. Now these cells are the first to be recruited at the sites of infection, and the cells are characterized by this multi-lobed nuclei, but scientists are not really sure why it's useful. Because neutrophils are a phagocyte, they produce hydrolytic lysozymes to digest substances. Now let's look at the macrophage, which you do need to know. This is the largest phagocyte in the body, and as its name suggests, it's a big eater. This cell, like the neutrophil, carries out phagocytosis, which we'll look at later on, but it has the ability to process and present antigen on its cell surface to activate T helper cells. And hence, this is called an antigen presenting cell, or APC for short. The T lymphocyte is named as it develops in the gland known as the thymus, which is next to the lungs. There are two main types of T lymphocytes. First up are the T helper cells, which coordinate the adaptive immune responses, which we'll look at later in this video. And next up are cytotoxic T cells, which are mediated by T helper cells to kill cells. And they typically kill virus-infected cells via a process called apoptosis, as shown in the GIF here. Another type of lymphocyte is the B lymphocyte, so called because it develops in the bone marrow. And B cells are covered by specific shaped antibodies, these Y-like structures. And once activated, B cells become plasma cells, which produce many monoclonal antibodies. Additionally, like the macrophage, the B lymphocyte also functions as an antigen-presenting cell, and we'll learn more about what this means later on. So, now there are two main types of immunity. First, we have the innate immune system, which we are born with, and it is not specific. This means it doesn't target one specific pathogen, but a whole variety. It's a bit like the police officer, as is the body's first-line defense. But you may wonder, why do we need the adaptive immune system, seeing that we already have a rapid protective system? But it's a bit like saying, why do we need special forces if we have the police, such as the MI5 or the army? So the adaptive immune system comes here, and it develops and trains throughout our life. Although it's slow, it's very specific to the pathogen, and why this is we'll see later on. So let's first look at the example of an innate immune response, phagocytosis, which is the process of eating cells. This is carried out by phagocytes, which is an umbrella term for all cells that eat cells, remember. And phagocytes are activated by signals released from T-lymphocytes or when they recognize foreign antigens, which is what happens more commonly. Once a foreign cell is identified, example bacteria, the phagocyte engulfs the bacteria, forming a phagosome. Then the lysosome fuses with this phagosome, forming a phagolysosome. A mouthful, isn't it? The lysosome releases hydrolytic lysozymes to digest the bacteria and then present the antigen of the pathogen on its cell surface. A common cell that carries out this process is the macrophage. So, after phagocytosis, the macrophage presents the antigens of the pathogen, and the specific T cell complementary to that antigen binds to it and becomes activated. Once it's activated, it can differentiate into either a helper T cell or a cytotoxic T cell. And this is how the innate immune system communicates with the adaptive immune system. The innate immune system presents antigens to the adaptive immune system. So the adaptive immune system consists of two types of lymphocytes. It might look like I'm reiterating here, but because this is a complex topic, this would help you. The T lymphocyte consists of the helper T cell and the cytotoxic T cell. And finally, we have the B lymphocyte. As explained before, T cell activation is needed for the adaptive immune system to function. However, activation is slow because finding the specific complementary T cell to the antigen of the pathogen is like finding a needle in the haystack, quite literally. Remember that T cells are activated by antigen-presenting cells, such as the macrophage or B cell, and once activated, they can carry out many tasks, which we'll see next. So the T lymphocytes carry out many functions, but more specifically, the T helper cells carry out the most. The T helper cells can recruit more antigen-specific cytotoxic T cell, they can upregulate phagocytosis, they can activate specific B cells, and also produce lymphokines, which aid in the cloning of B cells. Finally, the cytotoxic T cells that are produced can kill virus-infected cell via the process of apoptosis, which I've told previously. The B lymphocytes or B cells can be activated in two ways. One is if complementary antigen binds to the antibody that covers the B cell. Alternatively, a specific T helper cell can find that specific B cell, and this is called clonal selection. The activated B cells divide rapidly by mitosis to make plasma B cells, which produces many monoclonal antibodies. Now let's look at the details of the two mechanisms of how the adaptive immune response works. Remember, these are not types, these are the mechanism, the way they work. The cellular responses involves T cells activation mediated by the presentation of the antigen of the pathogen by macrophages. However, in a humoral response, there is B cell activation and antibody production. And the reason we need these different types of responses is because the way the pathogens infect us. Some pathogens, like the virus, invade cells, and so they are intracellular, so you would require a cellular response. While pathogens that infect extracellularly, common examples being bacteria, require a humoral response, an activation of B cell and antibody production. However, in an active infection, most likely both responses occur. However, one response will dominate over the other. So following an immune response, memory cells are made, which you may be familiar from GCSE. This is important because remembering the pathogen allows for a quicker and a stronger immune response on secondary exposure to the same pathogen. This is just like if you go through this video again, you're going to understand the information quicker and have a stronger knowledge. Not funny? Hey, I tried. Anyways, many monoclonal antibodies can be made quickly and pathogens can be killed before even causing symptoms. And generally, memory T cells remember the specific antigen of the pathogen, while memory B cell remembers the specific antibody complementary to the antigen. So here's a recap of the aims that we went through this tutorial. As usual, use them as questions to consolidate your learning. I thank you for watching, and I'll see you in the next one.